Assalamualaikum guys, today my topic of presentation is the different modes of transcription regulations in pursuit of design principles of regulatory sequences. Okay, so my table of content is as follows: introduction, regulatory building blocks, quantitative characterization of transcription factor binding sites, method used, and their regulatory architecture composition. Too many transition in this slide. Okay, so first we have to know what are the transcription factor binding sites. We know that the gene expression is controlled by many different enzymes, many different transcriptional enzymes, translational enzymes, and many different other activators, enhancers, and different cofactors are involved in this process. So transcription factor binding sites are the sequences to which the different transcription factors bind, and they tend to increase or decrease the transcription. It all depends upon their binding, their location in the genome, their number in the genome, and their different and the different architectures they form. So we partially know about the regulatory instructions encoded in the genes by the advanced technologies. Regulatory DNA sequences was discovered in 1960, and since that time the main focus was on the regulatory instructions are encoded in these sequences. Like regulatory sequences have the regulatory instructions in them. And what are those instructions actually? Those instructions are the binding of that sequences with their respective transcription factors. So any change to these sequences, any change to the binding method of the sequences with their transcription factors can lead to the change in transcription. It can lead to the increase, decrease, or stop of the transcription at all. And deviation from the desired expression patterns. What is deviation from the desired expression patterns? Like if a certain gene is overexpressed, it is down expressed, or it is abnormally expressed, it can lead to different diseases. For example, cancer. In cancer, there are many cancer controlling genes that are downregulated, and in many other conditions too. So what are the building blocks of these? What are the building blocks of those regulatory sequences which control the gene expression? Transcription factor binding sites are the building blocks for this one, and they usually have the short sequences of 6 to 12 base pairs. And these sequences have very distinct specificity for the respective molecule that are the transcription factors, and binding of these will further result in an increase or decrease expression.